secondary growth, and almost in the secondary growth we have talked a lot of things. Okay. Mm, Out of this, some of the important portion related to this secondary growth is that in the vascular portion we have actually learned yesterday. No? The activity of vascular cambium, is it? This activity of the vascular cambium, this produce secondary vascular tissue, that is secondary vascular tissue is secondary xylem and secondary phloem, is it? This secondary phloem is produced on the outer side and the secondary xylem is produced on inner side. This production of the secondary xylem and secondary phloem, the activity of cambium is more on the inner side. Activity of cambium is more on inner side, is it? So more secondary xylem is produced, and this is known as wood. Okay, so wood is produced, and less secondary phloem is produced. This activity is also influenced by the environmental condition and particularly by temperature. And so during this spring season, there is a production of spring wood. So this caused the production of spring wood, and during the winter and the autumn, there is production of autumn wood. And this spring wood and autumn wood together make the annual ring, is it? And so every year one ring for the secondary growth is produced, and so these annual rings are useful for calculating the age of the tree. Okay, another thing is that as the secondary growth takes place, naturally the more tissues are produced on the inner side, and the inner side the space is less, is it? There is a limit in the space, isn't it? So what happened? Gradually this cambial ring move outward as the tissue is produced on the inner side. So cambial ring gradually is pushed to the outward, and in the cambium tissue new cells are also added so that the cambium ring remain continuous, remain continuous. Okay, and at the same time another things happen. This those secondary xylem which is produced on the inner side. Of course, all the secondary xylem is produced on the inner side, but as time passes, no, the wall of this secondary xylem, wall of a secondary xylem that become thicker more, and there is also addition of this resin, tannin, then oils, etc., and also tyloses. These things are added, and that's why gradually the the vascular that xylem vessels, they are blocked. Actually their lumen is gradually blocked. So this old secondary, that older secondary xylem become dark in color, that is darker in color, and they gradually unable to conduct water. This secondary xylem, which is located towards the center of the stem, that one is known as heartwood. Okay, this heartwood is towards center of a stem, towards the center of a stem, and this heartwood is also known as duramen. Due to this thick wall of this secondary xylem tissue and the deposition of resin, tannin, oils, and tyloses, this heartwood portion, they become very strong, and it provides only mechanical strength. It cannot support the conduction of water. But those secondary tissue, secondary xylem, which have formed recently, that is a new secondary xylem. This secondary xylem is slightly lighter in color, and they are active in water and mineral conduction. This secondary xylem, which has formed recently, that is later formed secondary xylem, that is known as sapwood. So there is, in the older trees, we can get towards the center dark colored wood, and towards the periphery we get light color wood, and the light color wood is sapwood. As the time passes, that is the tree grows older, the sapwood gradually convert into heartwood. So sapwood, it is gradually converted into heartwood. So in more old is the tree, more heartwood is there. Okay, this conversion. All right. So older the tree, more will be the heartwood portion. Sapwood portion is not much, but it will be always towards the periphery. Sapwood is near the vascular cambium. Okay, sapwood is near vascular cambium, and heartwood is away from the vascular cambium. Another things related to this secondary growth. Okay, one thing is important thing is tyloses. Tyloses, these are actually tyloses are balloon-like structure which are produced within the lumen of xylem vessels. Say what happened actually, say this is the xylem vessel, all right, having very thick cell wall, and say here is these are the xylem parenchyma. This xylem parenchyma and this xylem vessels, they are connected with the plasmodesmata. Okay, there are pores, all right, perforation is there. So what happened? This is the living portion, this parenchyma, the plasma membrane from this parenchyma, xylem parenchyma gradually move inside the xylem vessels, and there it swells like a balloon. All right. And this balloon like structure is known as tyloses. So, tyloses are seen in the older vessels, and so due to this production of tyloses, the lumen of the xylem vessel is blocked, and so now it cannot translocate water through it. So the pipe is blocked, water pipe is blocked. So this is tyloses. One another thing is that the wood of angiosperm is hard and porous. Okay, hard it is due to in the wood of the angiosperm tree. What happened? They have xylem vessels. They have xylem vessels and they have lot of sclerenchyma also. Due to the sclerenchyma, it is very hard, and porous condition is due to presence of vessels. If you make a transverse section of the wood of angiosperms, then what you will find that the xylem vessel, which has a wide lumen, this under microscope, these wood appear like there are big holes in the wood, and so it is known as porous. So the xylem wood of the angiosperm is porous due to presence of vessel, and hard due to presence of both basal tracheids and sclerenchymatous tissue. But angiosperm wood is very soft. You know this pine tree, this wood is very soft wood, very easy to work with, and this is due to that they are soft because they have very less sclerenchyma. In their wood, only tracheids are there. Okay, only tracheids are there. Tracheids have very narrow lumen in compared to vessels. So there is, and these are closely packed. So this is non-porous, and the angiosperm wood is porous wood. So hard and soft, hardwood and soft sapwood, and the porous and non-porous wood. Next is that this was the secondary growth in the vascular region, in the stellar region. Secondary growth also takes place in the non-vascular portion. Say towards the center, this secondary xylem, secondary phloem is produced, but the peripheral portion where the cortex, epidermis and hypodermis is there, those portion also has to grow in their volume, okay, grow in their diameter, and this takes place by the cork cambium. So in the extrastellar region secondary growth takes place by cork cambium, activity of cork cambium, okay, and this cork cambium develop from either hypodermis or inner layer of cortex. So from the parenchyma or the collenchyma tissue that is present in the hypodermal region, that one convert into meristematic tissue and forms the cork cambium. Here in this portion, this is showing in the stellar region. The secondary growth here is in the green portion. Okay, here is written one is phellogen. This phellogen is the cork cambium. Okay, this one, this cork cambium here developing from permanent tissue, simple permanent tissue. And so this cambium or this meristem is totally secondary in origin. All right, their origin is secondary in origin. This cork cambium tissue also produce cork cambium also produce secondary tissues, but they produce secondary non-vascular tissue. Vascular cambium produce secondary vascular tissue, but this portion is non-vascular. So this cork cambium produce non-vascular tissue. It produce on the outer side produce phloem. This is also known as cork. Okay, and on the inner side it produce this one. It produce phellogen. Phellogen, or we call it secondary cortex. Okay, phellogen or secondary cortex. Cork tissues, they are actually form of their cell wall, they are suberized, a waxy material, and due to presence of suberin in the cork tissue, there is suberin, and due to presence of suberin, it is impermeable to water. So water loss is checked due to presence of the cork tissue. On the inner side, it produces sorry, sorry, here this one is um, here on the inner side, secondary cortex. It is not phallogen, it is phalloderm. Phalloderm or secondary cortex. Phallogen is the cork cambium. So phallogen produce phloem and phalloderm. All right. Phalloderm is the parenchymatous tissue, but they are usually thick wall. Okay. This so here is cork cambium. Cork cambium. It is outer side is produce cork and inner secondary cortex. This one is phallogen and this is phloem and this one is phalloderm. This one, these secondary tissues, they produce a a very thick wall-like structure, okay, impermeable to water, and this portion that the phallogen plus phloem plus phalloderm derm together form a structure that is known as periderm. Okay, this three together is called periderm. In some places, this phallogen on this skin layer, this phallogen in certain places produce loosely arranged cell. Loosely arranged cells, and here the epidermis also breaks. These become natural opening on the bark of the tree, and this one is known as lenticel. Lenticels or lenticles. These are actually natural opening through which they help in the transpiration and also help in gaseous exchange. Uh, no, no. আরে ইয়েটা একটা বইটা খুলছিল না। যা here I'll show you the structure of that lenticel. Okay. Here, this structure you can see. Hello, hello. Can you hear me?
Yes, sir.
Okay, this structure you can see. This
Yes, sir.
diagram from the book
Yes,
page.
sir.
Book page. This is the lenticel. Okay, here you can, I think, see that that epidermis portion has been broken. Okay, and there is complementary cells. These are loosely arranged cells which is produced by the cork cambium. This cork cambium has produced the loosely arranged cells, and this portion, due to this loosely arranged cells, they can they can allow the release of water in the water vapor, and also gas etc. can be exchanged. And these are, say, the secondary cortex. Here the cork cambium you can see, but at the place where this lenticel is produced, this the cells are loose, and cork cambium also you can see what type of, that very loosely arranged. This one is showing the bark portion of the tree, that due to continuous growth of the formation of the secondary tissues in the tree, and increase in their diameter, the bark portion, older part of the bark, it cracks and it come out gradually. So this is the all related to the secondary growth. Now let us see some of the questions based on the entire anatomy. Okay, anything you have to ask from here? Do you have anything to ask? Hello?
No, sir. No, sir.
No, nothing. Okay, then we will now we will see some question based on this anatomy. Okay. Here is our the first question. This one, the lignified cell wall occurs in, the options are epidermal cell, phloem cells, xylem cell, and cambium cell. Which has lignified cells? Can you tell which has the lignified cells? The cells with lignin. Cells with lignin on their wall are sclerenchyma. Sclerenchyma is with lignin. Tracheids are with lignin. and the vessels, xylem vessels are with lignin. Okay, so here the options, our options are epidermal cell, phloem cell, xylem cells, and cambium cell. So here it is the xylem cells. Xylem, main component of the xylem, the details has not given. Xylem is made of tracheids and vessels. So xylem is the answer. So xylem cells are lignified cells, which is enucleated at maturity. That is, cells without nucleus. Cells without nucleus, so which one? Here is sheep cells, companion cells, cortical cells, and palisade cell. Palisade is palisade, that is the mesophyll of. Palisade is what? Mesophyll of leaf, dicot leaf particularly, dicot leaves, and cortical cells, that is cortex, they are also parenchyma. Parenchyma, they have nucleus. Companion cell which is attached to the seed tube. Companion cells are, they are closely attached to seed tubes, and they have very prominent nucleus. Only it is the sheep cells or the seed tube elements. No, sheep tube elements, they don't have nucleus at maturity. Tube elements are without nucleus. At maturity only. In the earliest stage, actually, they have nucleus. See, nucleus is absent in sclerenchyma also, tracheids also, vessels also. But actually, these are the dead cells. This sclerenchyma, when they are mature, they are dead. So naturally, they don't have nucleus, but they are dead. But here, the sheep tube cells, they are living. They are living with living cytoplasm, but still they don't have nucleus. Next one, come to that healing of wounds occur due to the activity of healing. Healing of the wounds, the injured portion, is done by, how it is done? It is done by adding new cell. New cells are added. Addition of new cells cause healing. So naturally, injured portion or wounds can take place in any part of the body, and in those part of the body there is permanent tissue. There is permanent tissue, and permanent tissue, they do not do not uh, divide, they do not produce the new cells. So what happen, if there is anywhere there is wound, the cells in the neighborhood of that wound, okay, permanent cell, if there are living tissue is there, okay, those permanent living cells, say In a injured portion, parenchyma tissue is there, collenchyma tissue is there. Those cells, they become meristematic. They become meristematic. So meristem is developing from permanent tissue, and the meristem which develop from permanent tissue, that is the secondary meristem. So healing of wounds takes place by secondary meristem. Anywhere, once the healing is completed, then those meristematic tissue again change into permanent tissue. This process is called the is called de-differentiation, de-differentiation and redifferentiation. Okay, by de-differentiation, permanent tissue is differentiated. So remember that permanent tissue is formed by the process of differentiation, and from this differentiation they have to come back to meristematic. So that one is de-differentiation, means it is moving in the opposite direction again, coming back to its normal initial position. And once it is work is done, then those meristematic tissue cells, they go back to again permanent tissue, means again differentiation takes place. As the differentiation is taking place second time, so this is known as redifferentiation. All right. Next, this is a matching. Match the following and choose the correct option. Here in this column 1, this side is column 1 and column 2. Okay, column 1 and column 2. Column 1, we have meristem, parenchyma, collenchyma, sclerenchyma, and epidermal tissue. In the column 2, we have photosynthesis synthesis and storage. Next, mechanical support, then actively dividing cells, astomata, and sclerids. So here, what will be the matching? See, meristem is actively dividing cells. All right. And parenchyma, what is the function of the parenchyma? Here, actually, these two columns. In one column, it is the type of cells or tissue, and on the other side, in the column 2, it is the function it carries. So, meristem is actively dividing cells, that is the characteristics. Parenchyma carry out photosynthesis and storage. When the parenchyma tissue is with chlorophyll, so they carry out photosynthesis, that is the chlorenchyma. Is it? Chlorenchyma, they carry out photosynthesis, and also the other parenchyma carry out storage. Then collenchyma can provide mechanical support. Actually, collenchyma is present in the hypodermis. You know, hypodermis of the dicotyledonous stem, right? And in the young branches, young stem, there is no vascular tissue. So this collenchyma, they can provide some mechanical support because their wall is thicker than the parenchyma. Though it is irregularly thickened, it can provide some mechanical support. Then sclerenchyma here will be matched with sclerids. Sclerid is a type of sclerenchyma tissue. Sclerenchyma tissue are two types. One is fiber and another one is sclerids. Is it? So this is type. So sclerenchyma can match with vessels. And epidermal tissue here will match with stomata because stomata is present in the epidermal tissue system. So stomata is those guard cells. Guard cells are modified epidermal cells. So it come under epidermal tissue. So the matching, which one will be our answer? Here it is means A with 3. A with 3 is this one. B, option B. There is no one else is A with 3. So directly from here we can find out that this is the only option where it will match. Next question. Which of the following is made up of dead cells? Made of dead cells. Here options are collenchyma, phloem, and xylem parenchyma. Which one is dead? See, xylem parenchyma, in the xylem tissue, xylem parenchyma is the only living cells. This one, xylem parenchyma, is living. In the phloem also is living. Sheep tube, companion cell, phloem parenchyma, they are living. Where there is a fiber, phloem fiber, that one is dead. The collenchyma is a living tissue, living cells. The phloem, which is also known as cork, and cork is a dead tissue, okay, which forms the outer portion of the bark. All right. So this one is cork is dead. Cork cells are produced, their cytoplasm is lost, and there is suberin is produced in their cell. Suberin deposit is there. So they are suberized cell. Cork is suberized and it is dead. So phloem, it may be, so this one, option 2, phloem is the answer. Next question, again matching with column 1 and column 2. Here this side some of the structure in the column 1, and in the column 2 their properties and some function etc. has been given. So here in the column 1, cuticle, bulliform cells, stomata, epidermis. And in column 2, guard cells, single layer, Vascular empty colorless cells. So where it can match? Cuticle. Cuticle is actually, the cuticle is present on the outer surface of the epidermis. This is a layer on the outer surface of epidermis of all the aerial part of plant. In submerged aquatic plant and underground part of the plant, they don't have cuticle. Okay, so this is a waxy layer. This is made of cutin. Cutin is the chemical. This is waxy nature, wax like. Bulliform cells. Bulliform cells are which is found in grasses, grass epidermis. Grass and some other plants. This is bulliform cell is found in grass epidermis. Due to these are large cell, they are empty and colorless. A specialized epidermal cell of the leaf, and due to change in the turgidity of the cell, like the guard cell of the astomata, this cell also, due to their turgidity change, when the cells are turgid, leaf is widely open, and when the cell become flaccid due to water stress, the cell flaccid means their volume is decreased, and at that time the leaf will curl inward, so they will reduce the water loss. This is an adaptation to the xerophyte, a adapted measure for the xerophyte. Next is astomata. Astomata is made of guard cell. Okay, and then epidermis. Epidermis is most of the cases single layer. Is it single layer of cells? So epidermis is single layer. In some of the xerophyte, the epidermis is a double layer. Okay, those the double layer epidermis is in only a few xerophytes. So it will match with what? It will match A with 3 is there. Say in the option when we will see A with 3, here it is difficult. You cannot decide immediately if you don't know all. So number 1 option is A with 3, number 3 option also A with 3, number 4 also A with 3. But if we see B, bulliform cells should be with 4. So this one B with 4 is present only in option 1. So naturally in this one is the answer. Others will match naturally. Say automatic they will match. C is 1, astomata, guard cell 1, and D with 2, that is D, epidermis is single layer 2. So this is answer. Next question come to the monocot root differ from dicot root in having monocot root differ in dicot root in having a scattered vascular bundle. This one is, say this is character of the monocot stem, not with the root. Well developed pith, say in monocot pith is well developed. Large pith is there. The monocot, I think I told, pith is prominent pith. Then polyarch condition, that is xylem bundles, xylem and phloem bundle greater than 6, and this is mainly these two will give the difference between the dicot root and monocot root. Say this three radially arranged vascular bundle. The radial vascular bundle, it is characteristics of common character of root. So it is present in monocot as well as in dicot. So this cannot be the differentiating point of the monocot and dicot root. Open vascular bundle. Open vascular bundle is, this is the characteristics of dicot stem. Say last question one. Conjoined collateral and closed vascular bundles are found in conjoined collateral and closed vascular bundle. It is the characteristics of monocot stem. Okay, but here in the option they have not written monocot. Dicot directly not written. Here it is given grass root, grass stem, sunflower stem, cucurbita stem. Now this is closed vascular bundle in monocot stem. So here sunflower is, it is a dicot. Cucurbita is also dicot. Grass stem, this one is monocot. It is not the character of root. So this our answer will be here grass stem. Okay. So today up to here, do you have anything? You can at home also, you can practice from your practice book questions and can solve uh, different questions from different years questions which were asked in the NEET exam. Okay, I have some question from the different years of NEET. Those things, but in next class we can do it. Some more question. Next class also we will discuss questions only. Okay. So thank you. Do you have anything to ask? Any query? Any
Sir,
question? Yes.
can you explain again that bit and
Yes.
falling
Yes.
out? What that question? That
I can. I
bit,
cannot.
sir.
Some sound is
The
noise. Yes.
above question, sir.
Other
That poly art. Can
poly.
you explain again? Yes, sir. Yes.
See, when the vascular bundles, no, this the in root or stems also, the in the root there is a usually 2 to 4 that xylem and phloem bundles. All right. In some cases, there is only one also. So when it is two patches of xylem and phloem bundle, then we call it is a di-arch. Di means two. If it is So when 2, it is di. If it is 3, we can call it triarch. Okay, means 3 xylem bundles are there. If it is 4, we can call tetrarch, right? Like this way, pentarch. If it is 6 xylem and phloem bundles alternately, then we can call them hexarch, is it? So when usually what happen, if it is 2 to 3, then we can 2 to 4, then we can call them that diarch or tetrarch or triarch. But when it goes many, polyarch means when it is many xylem and phloem bundles, then we call it polyarch. In case of monocot, monocot root, there is more than 6 xylem and phloem, xylem and phloem bundles. So this condition we can call that they are polyarch. Poly means many. Okay, clear?
Yes, sir.
I can show you some diagram from the book itself. You can see this. You can see your book here, book pages coming on the screen. Say this is the this is transverse section of the root. Okay, you can see it. Hello,
Yes, sir.
you can see the diagram of the root.
Yes, sir.
You can see how many xylem you can count. 1, 2, 3, 4, 5, 6, 7, 8. 8 patches of xylem, and alternating with that there is phloem. These portions are phloem. Okay, phloems are here. The small cells are there, no? So these are phloem. So this condition is polyarch condition. Okay, and this one is a dicot. Here you can see how many? 1, 2, 3, 4. So this is a tetrarch condition. Okay, so this is polyarch and tetrarch or pentarch. Any other question from anyone? Any other? Okay, you practice at home with your practice book and try to solve question. Whatever question comes